Good day everyone, we are the group 3 and for today we are going to present one of the microbial infections of oral cavity, which is the cytomegalovirus. We're going to be talking about or discussing the etiology, structure, pathophysiology, clinical manifestations, and histologic examinations of CMV or the cytomegalovirus. So CMV or cytomegalovirus is a part of a big family of viruses called herpes viridae. It is important to note that the subspecies of herpes viridae called beta herpes virus 5 is the one that infects the human body. Thus, it is an infection that is common, and once the body is infected, the virus will likely remain there for life. And it can be either dormant or active for some people. This is why most patients from across the globe that suffer from this disease don't actually know they have CMV unless they are diagnosed with it or they know firsthand, because not only is it likely for life, but it also causes problems very rarely in healthy people, which is what you call very dormant for them. However, if for example a person has a weak immune system, the virus activates or reactivates and can have very fatal results. And the etiology is that when the virus is active, it can be passed through bodily fluids and can also from pregnant women to their babies. And now let's move on to the structure of the organism. Please refer to the picture shown for this slide. The cytomegalovirus is a member of the beta herpes subgroup of the herpes virus family. Its structure consists of an enveloped outer lipid bilayer that is made up of different kinds of viral glycoprotein. CMV has a spherical shape with an icosahedral symmetry. So if you're not familiar with the term icosahedral or what icosahedral is, it is basically a polyhedron with 20 faces. The icosahedral protein capsid of CMV has an average diameter of 100 and is made up of 162 hollow hexagonal and pentagonal capsomeres with an electron-dense core holding a 230 kbp double-stranded DNA genome which forms the nucleocapsid. Outside the capsid of mature virus particles is an amorphous proteinaceous layer known as the tegument, which is surrounded by a lipid envelope produced from host cell membranes. There are at least 25 proteins found in the tegument layer, so take note of that. The tegument is made up of enzymes such as VP16, which is in charge of subverting cellular proteins and enzymes involved in viral nucleic acid replication, and VHS, or virion host shutoff, which is responsible for shutting off host cell protein synthesis in the cytoplasm. One of the main CMV surface glycoproteins, GB, affects the virus's ability to bind to and enter cells as well as the spread of the virus from cell to cell. Another relatively common glycoprotein of CMV is GH, which serves as a target for complement-independent neutralizing antibodies. Okay, for the pathophysiology, the lytic virus or cytomegalovirus CMV has a cytopathic effect both in vitro and in vivo. Once a person gets infected with this virus, it will stay in their bodies forever. So just like I said from the second slide or from the etiology, once you are infected by this virus, it will likely remain for life. The bodily fluids such as blood, urine, Even breast milk from a pregnant woman or a mother, semen, and saliva are some ways to transmit cytomegalovirus to another person. All the organs here in the body will be affected by this one because of it likely spreading in the bloodstream. But it will only be clinically obvious once the levels of the virus KOAD increases. So people who have a weak immune system may experience this virus more fatally than others, especially those who have undergone bone marrow, organ, and stem cell transplant. So if you're very, very unlucky, you can experience this all at once. Weaker immune system, experiencing the CMV, and undergone transplant, may it be bone marrow, organ, and stem cell. So if you're in that stage, it can be very fatal and dangerous. Clinical manifestations. Cytomegalovirus causes only minor illnesses in healthy person. On the other hand, people with compromised immune systems may develop CMV mononucleosis, a disease in which there is an excess of white blood cells with a single nucleus. The most typical symptom of CMV illness in persons with HIV is retinitis. Patients with CD4 counts less than 50 cells per liter are more likely to experience it. Rates in this cohort can reach 40%. Patients with this condition describe floaters, loss of one side's visual fields, and diminished visual acuity. Thus, a typical virus is a cytomegalovirus. When a person gets the infection, the microbe will remain for all time. Again, I'm gonna be mentioning this. Most people are unaware that they have CMV because it rarely creates issues in healthy individuals. CMV should be avoided if you're pregnant or have a compromised immune system. But if you're born with it, or if you've acquired it from your mother, it is, again, you cannot avoid it. And babies born to mothers who become actively infected with CMV during pregnancy may later exhibit symptoms. Here we have pictures here for the signs and symptoms. You can see painful throat, swollen glands, enlarged tonsils, nausea, hepatitis, splenomegaly. Now, symptoms include, again, a painful throat, swollen glands, enlarged tonsils, fatigue, and nausea. It can induce liver inflammation or hepatitis, as well as spleen enlargement. CMV mononucleosis is comparable to Epstein-Barr virus mononucleosis, which is also part of the herpes family, in which CMV is also a part of. Now, the last part for this topic is the histologic examination. Endothelial cells lining tiny arteries are noticeably expanded in cases of cutaneous CMV infection. So you can see on the figure 1, or first picture, that the lining of tiny arteries are expanded. It can be seen na expanded siya. Next, when compared to normal or reactive endothelial cells, these are bigger and eosinophilic. A large, heavily eosinophilic intranuclear inclusion is the distinguishing histologic characteristic. So see second picture. It appears to be large and bigger. Here you can see on the arrow. Less frequently, other cells like fibroblasts and epithelial cells are implicated. The inclusions could be observed alongside nonspecific inflammatory alterations and underlying epidermal ulceration. A response pattern called leukocytoclastic vasculitis is identified. C4, And for the third picture, a highly specific method of establishing the presence of CMV is through immunohistochemical investigations against the virus. PCR is also used to detect CMV in a variety of clinical specimens. So that is all for our topic. I hope you learned something. Thank you so much and have a good day.